We've talked a bit about ATP and cellular energy, but up until now, we haven't talked about how that energy is utilized and stored. In this chapter, we're going to dive into what it means for cells to have energy and to do work. The language of energy is the language of physics and chemistry. Here, I will discuss this topic from a biological perspective and show how all life, including yourself, uses energy from the bonds of ATP. There are two types of energy: kinetic energy and potential energy. Previously, we have discussed kinetic energy. This is the energy of motion. Things moving around have kinetic energy. Kinetic energy is also called thermal energy when it is associated with molecular motion, but it is still the energy of movement. The other kind of energy is potential energy. This is the energy that something has because of its location or structure. If you pick a ball up off the ground and put it on the table, you have created potential energy in the ball. If we just magically remove the table, the ball will fall to the ground, releasing that potential energy. Atomic bonds also store potential energy. This is called chemical energy, but that just means it's the amount of energy within molecules that can be released as part of a chemical reaction. Glucose has more energy than CO2 held in its bonds. If we break glucose down into as many CO2 molecules as we can, the total energy in all that CO2 is less than the energy in the glucose molecule bonds. Where does that extra energy go? This brings us to the first law of thermodynamics. This law states that the energy of the universe is constant, which means that energy can be transferred or changed, but it cannot be destroyed. Biological organisms take in nutrients and convert the energy from their chemical bonds into forms usable by that organism's cells. However, the organism did not create energy. It simply transferred and changed it. When animals eat, that food contains sugars which their body can break down into CO2 and water. This releases energy. Some of this energy goes to fueling our bodies, and some of it is released as heat. None of it is destroyed. However, once the energy is lost to heat, it can no longer be used to fuel the body. This heat energy was transferred to the molecules in the atmosphere, and they passed it around between each other until the small amount of heat energy was being shared as kinetic energy by all the atoms in the environment. This leads us to the second law of thermodynamics. Since energy cannot be destroyed, this law states that energy transfer increases the entropy of the universe. Well, what the heck is entropy? Entropy is a fancy chemistry word for the disorder of a system. When energy is converted into heat, it is spread out and no longer can be used for doing work. So when a body produces heat and transfers it to the atoms in the air, they spread that heat out to so many molecules that even though the energy isn't lost, it becomes unusable. You can use the water from a hose to wash your car, but you cannot use the dewdrops from a field because it would require more work to collect them than is available. The water is still there, but it is not helpful because it is too disordered. This is what heat energy is like. We measure the amount of usable energy in a system with this symbol. The amount of energy stored in glucose is some value of G. This is called free energy. If we break glucose down into CO2 using this chemical reaction, each of those CO2 molecules has some amount of energy as well. We can subtract the total amount of energy in glucose and oxygen from the total amount of energy in the CO2 and the water. What we find is that there is more energy in the bonds of glucose and oxygen than in the bonds of CO2 and water. Thus, the change in energy, or the delta G, is negative. Reactions with a negative delta G are called spontaneous. It is a slightly confusing term, but it means that the reaction gives off energy instead of taking it in. The idea that chemical reactions give off energy, or use up energy, is reflected better in the terms exergonic and endergonic. From the Greek, exer is a prefix meaning out of, and ender is a prefix meaning into. Exergonic means that energy comes out of the reaction. And endergonic means that energy is required to be put into the reaction. In simple terms, exergonic reactions have energy as a product, and endergonic reactions have energy as a reactant. So, we have some reactions that are spontaneous, that is, they give off energy. And we have other reactions that are non-spontaneous, that is, they take in energy. The craftiness of living organisms is that they have evolved to link these two processes together. In other words, they use the energy given off in exergonic reactions as fuel to help power the endergonic reactions. This happens at the level of the organism. Even within your own body, there are exergonic processes being used to produce energy for endergonic ones. These are called catabolism and anabolism. We've heard a lot of words so far. Let's take a second to collect them. There are two kinds of reactions to remember: spontaneous and non-spontaneous. Spontaneous reactions are exergonic, which means that they have energy as a product. As part of your metabolism, we call these catabolic reactions. On the other side, we have non-spontaneous reactions. These reactions require energy as part of the reactant side and are called endergonic. As part of your metabolism, they are called anabolic reactions. So, when your body breaks down things like glucose, it can use that energy to make things like non-essential amino acids and lipids. This is called energy coupling. This brings us to the guest of honor: adenosine triphosphate, or as it is abbreviated, ATP. ATP is the link between catabolism and anabolism. The bonds in ATP have a lot of stored energy in them, and thus can donate that energy to endergonic reactions to allow them to happen. It is important to understand the structure of ATP, because as we have seen, structure is related to function. ATP is related to the structure of DNA. It has a ribose sugar, a nitrogenous base adenosine, and a phosphate group. However, this structure also includes two more phosphate groups attached to the end. This is why it is called triphosphate, because tri means three. The last phosphate functional group has a lot of energy contained in its bonds, but the negative charges on the other two are repelling it, so it is easy to pop off. Here's what the reaction looks like. ATP plus water produces ADP, a phosphate group, and energy. As we've seen, this is a hydrolysis reaction. So how does this help the cell? If the phosphate group just pops off, the energy would be lost to the environment as heat. The answer is that most of the reactions in your body use this energy by transferring the phosphate group. We saw this type of transfer in the pharmacology section last time when we looked at kinases. We've also seen active transporters, which use energy that they get from ATP to move molecules up their concentration gradient. We can now see how this is done. The third phosphate from an ATP molecule is transferred to the transport protein, which causes it to change shape. This is called a conformational shift. This shift also cleaves the phosphate and allows the protein to reset. Thus, the transporter, which is an endergonic reaction, has used the exergonic reaction of ATP hydrolysis to do work, to push these molecules up their concentration gradient. In a day, your cells combined use up about your entire body weight's worth of ATP. Where does all this ATP come from? Remember the mitochondria? They are the energy factories of the cell. They use the catabolic or exergonic reactions that break glucose into CO2 to fuel the anabolic or endergonic reaction of making ATP. We will look at that process more later on. But what is important to know now is that the exergonic reactions like glucose metabolism and the endergonic reactions of transport proteins in the membrane are linked through this amazing molecule, ATP. The catabolic and anabolic reactions that take place in your cells utilize proteins to speed up their reaction time. They do this because they are a catalyst. Catalysts are molecules that help speed up a chemical reaction, but are not consumed as part of the reaction. Normally, even spontaneous reactions take long periods of time to occur. Proteins that speed up chemical reactions are called enzymes. We saw kinases last time, which are enzymes that sped up the addition of phosphate groups to other proteins. enzymes increase the rate of chemical reactions. This helps your body keep pace with all the reactions necessary for life. Some molecular products are made by the cooperation of many enzymes in a sequence. This is called a pathway. Like we saw last time with the phosphorylation cascade, each kinase is an enzyme that contributes a little bit to the final product or response. Enzymes work by binding to substrates, which can be small molecules or even other proteins. These substrates bind into what is called the active site of the enzyme. This is a physical location on the enzyme that fits the substrate and helps bring the reactants into close proximity, and then helps them rearrange. This lowers the activation energy of the reaction and makes it go much faster. Even in exergonic reactions, it takes a little bit of energy to get the bonds to break so that the reaction can proceed. Enzymes provide this by interacting with the reactants. The side chains of amino acids that make up the protein help lower the activation energy and speed the reaction up. Some enzymes require cofactors or coenzymes to help them perform their actions. These are things like magnesium and vitamin C. Vitamin C is a coenzyme in the reaction to make collagen. Without it, you would get scurvy. However, taking vitamin C over the needed amount does not make the reaction go even faster. Vitamin C is only a coenzyme and can't work faster than the enzyme's own speed limit. While some small molecules can help enzymes do their job, others can inhibit them from their work. Surprisingly, these are called inhibitors. Many pharmaceutical drugs are enzyme inhibitors. There are two basic types of inhibitors: competitive inhibitors and non-competitive inhibitors. The name competitive inhibitor tells you what it does. It competes for the active site on the enzyme. Competitive inhibitors often have the same or a similar shape as the reactants. But once they're in the active site, they don't react. They just block the site from receiving the real reactants. An example of competitive inhibitors are statins. Statins are drugs that help block the production of excess cholesterol by competitively binding to the active site of HMG-CoA reductase, the main enzyme responsible for cholesterol synthesis. Other molecules can inhibit the enzyme through non-competitive binding. In this case, the molecules bind to some other place on the enzyme, but not the active site. This binding causes the enzyme to change to a new shape that blocks the active site. Once again, this is a conformational shift. An example of a non-competitive inhibitor is penicillin, which inhibits the bacterial enzyme DD transpeptidase and prevents the formation of bacterial cell walls, allowing for your body to more effectively kill them. Non-competitive inhibition is also called allosteric inhibition. Allo means different, as in different from the active site, and steric means shape or arrangement of the atoms. Allosteric means a change in shape from somewhere other than the active site. Sometimes, allosteric changes can help an enzyme. In the hemoglobin molecule, each oxygen that binds opens up the enzyme to bind more oxygen molecules. In fact, it can bind a total of 4 molecules, and each one increases the efficiency of the next. In the case of hemoglobin, this could be considered a positive feedback loop. As each oxygen binds, it increases the binding of more oxygens. Positive feedback loops are rare, because they can run amok. However, negative feedback loops are common. In biology, feedback loops are frequently the product of a molecule allosterically binding to an enzyme that helped make it, to slow down its own production. This keeps the body from making too much. An example of this is the production of, you guessed it, ATP. ATP production is important and is tied to glucose metabolism by a long series of enzymes, which we will look at in a later chapter. However, if too much ATP is around, it will bind to one of the first of these enzymes, phosphofructokinase. This causes allosteric, non-active site binding that causes a conformational shift or a change in shape that produces inhibition and slows the production of ATP. All living things need energy to help drive endergonic reactions. We have seen that they use enzymes to couple endergonic reactions with exergonic reactions. This allows for the exergonic breakdown of food to help fuel the production of cellular components and necessary molecules.